Okay, since 65% or roughly 65% of our body is composed of water, we should probably talk a little bit more about water and some other biomolecules. Water's not what I would consider a biomolecule, but it is certainly the most ubiquitous molecule in our body. Once again, for review, water is H2O, 2 hydrogens, 1 oxygen. Water is also a polar molecule. And as such, there's a positive end to the molecule due to the electrons being pulled towards the oxygen atom, which is very electronegative. The oxygen atom has a partial negative charge. That partial negative charge is canceled out by the partial positive charges on hydrogen. So this is water. These are water molecules. If we look over here, this is a glass of water, or you could just assume that this is the human body with water in it. Water is the solvent. Water is considered the universal solvent within human biology, within all of animal biology for that matter. Water's the solvent. Anything suspended in the water is what we refer to as a solute. So in this case, we have glucose suspended in this water. We have sodium suspended in the water, potassium bicarbonate, and that's really all we have in this water right here. So these are all functioning as solutes. Certainly, all of these things, glucose, sodium, potassium, and bicarbonate, all have their distinctive roles in the body. But when they are in solution, that is to say, within the solvent of H2O, they're functioning as a solute. The measurement of the solute concentration within our body is known as osmolarity. Once again, osmolarity is the measurement of the solute concentration of a solution, whether that's within a cell, outside of cell, within a specific tissue. And once again, that is based upon the number of solutes in relation to the volume of water. It doesn't matter what solute we're talking about. That is to say, this could be all glucose molecules within this water, or it could be all bicarbonate. But the reality is, within our fluid, within the extracellular fluid, intracellular fluid, cerebral spinal fluid, any of the fluids in the body are going to have a litany of different solutes suspended within that water. So osmolarity is counting or relating the total number of solutes to the volume of water. Now, if we go back to this image right here with these water molecules, because water is polar, that is to say it has a positive region and a negative region, the positive regions and negative regions of adjacent water molecules tend to be attracted to each other because opposite charges are attracted to each other. So for example, this oxygen right here, which has a partial negative charge to it, is attracted to this hydrogen, which has a partial positive charge. And this is representing what we refer to as a hydrogen bond. Water molecules engage in hydrogen bonding with other water molecules. So this oxygen will form a hydrogen bond with this hydrogen. And to be clear, I said that water will form hydrogen bonds with other water molecules. That is certainly true, but hydrogen bonding occurs between hydrogen atoms, say, that are bound within hydroxide, which is OH negative officially, to other molecules. And we'll talk about this when we talk about proteins. The shape of proteins is very influenced by hydrogen bonding. So going back to this example here, these hydrogens and oxygens are attracted to each other due to their difference in charge. And this is creating what's known as hydrogen bonding. Okay, so one thing we should point out, or one thing I should point out, is that because water is polar, it gets along well with other polar molecules or charged atoms. Once again, charged atoms are known as ions. Positively charged atoms are cations. negatively charged atoms or anions. And this was not intentional, but all of these solutes within the water mixed well with this water because they are either polar, in the case of glucose, or charged in the case of sodium, potassium, and bicarbonate. To be clear, bicarbonate is a molecule, or more specifically, a molecular anion, whereas sodium is merely an ion, it's a charged atom. So to be clear, hydrophilic means water-loving. So all polar molecules tend to be, or let me rephrase this, all polar molecules are hydrophilic. They love being around water. They get along with water, they can hang out with each other without a problem. All polar molecules, which are hydrophilic, are also lipophobic. That is to say, they don't get along or mix well with lipid-based molecules, fat-based molecules, steroid molecules, phospholipids. Really, when I say phospholipids, I'm specifically saying the lipid component. Hydrophobic is describing a lipid-based, fat-based, steroid-based molecule that doesn't enjoy being around polar molecules or doesn't enjoy being around water. Something that is hydrophobic is naturally going to be lipophilic. That is to say, they enjoy hanging out with other lipids. Back to this right here. This hydrogen bonding will create what's known as surface tension, where the molecules are causing water to stick together. Pretend these are microscope slides or panes of glass, and I'm going to dip them in that imaginary water. These are totally wet. You put these together, you can easily slide these pieces of glass upon each other, but it's very, very difficult to separate these slides or these panes of glass due to the surface tension created by the water molecules. Even Dwayne "The Rock" Johnson would be challenged to actually pull apart these panes of glass due to this very strong surface tension. And we can see a great example of that within serous membranes. Specifically, right here, we have a serous membrane around the lungs. This is a lung, it's the right lung, but it doesn't really matter for this example. Inferior, below the lung, we have the diaphragm, in red right here. In blue around the lung, we have the serous membrane. We have the parietal pleura and the visceral pleura. The visceral pleura is the blue line directly adhering to the lung, and the outer line is the parietal pleura. To be very, very clear, these are the exact same line. If you were to follow this blue line all the way around, you would notice that you never have to pick your finger up because it's the same line. But what's important here is that there's a space between the parietal and visceral pleura known as the pleural space. It's somewhat of a misunderstanding when people hear the word pleural cavity, that's the cavity that the lungs reside in. The lungs actually reside in the thoracic cavity. The pleural cavity is merely the space between the parietal and visceral pleura. Once again, these are serous membranes. Within that space is serous fluid, and in this case, we would call it pleural fluid. And it causes these membranes, the parietal and visceral pleura, to stick together. So if I was really going to draw this more accurately, it would be just one blue line because the visceral pleura and parietal pleura stick together. I've separated them just so I can demonstrate that there's actually two membranes. But the reason they're stuck together is due to the surface tension of the water molecules within the serous fluid or the pleural fluid, causing those membranes to stick together. Why do we want the membranes to stick together? Because if they don't, we cannot breathe. Our lungs get larger and smaller, and they do that with changes in shape of our thoracic cavity. As our thoracic cavity gets larger, our lungs get larger. And when our lungs get larger, the volume of the lungs gets larger, pressure decreases, so atmospheric air can then flow into our lungs. We don't breathe in air and cause our lungs to get larger or our thoracic cavity to get larger It's the antithesis of that. When our muscles of our rib cage and of our diaphragm pull our lungs to make them larger, the increased volume of our lungs is decreasing pressure so much in the lungs that air flows into our lungs down a pressure gradient. And we'll talk more about that when we get to the respiratory system. But this would not be possible without the surface tension within the serous fluid. Our lungs, or let me rephrase this, our ribs, attached to the inner lining of the ribs is the parietal pleura. When our muscles take our ribs and pull them outwards, it's going to take the parietal pleura with it. That's the outermost layer right here. And because the parietal pleura is physically stuck to the visceral pleura is directly adhering to the lung. When the ribs move outwards or the diaphragm moves down, those structures are pulling the lungs outward and inferior. And because the visceral pleura is directly attached to the parietal pleura, it makes the lungs larger. And this would not be able to happen without the surface tension created by the water molecules. Let's talk about some biomolecules. Carbohydrates, proteins, fats, and nucleic acids or nucleotides we'll touch on. The main carbohydrate that we're concerned with within human physiology is glucose, C6H12O6. And this means that there are 6 carbons, 12 hydrogens, and 6 oxygens. So let's just look at the carbon atoms. Every vertex within this molecule or with any carbohydrate molecule that is drawn, the molecular shape of the molecule, when we see a vertex like this here or these points, that's representing a carbon atom. So if we count these, this is the carbon atom number 1. And there's the 6 carbon atoms. 1, 2, 3, 4, 5, and 6. C6H12O6. Now, we don't see all the hydrogens. A lot of the hydrogens are assumed that they're there. Carbon atoms prefer to have 4 bonds to them. So this carbon atom has this bond right here, this bond right here, that purple bond going to the OH group right here, and then there's an implied hydrogen coming off of here. So if we wanted to count the hydrogen, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Glucose is a monosaccharide, meaning it is just one glucose molecule. There are 2 other monosaccharides that make up carbohydrates. They are galactose and fructose. Galactose looks very, 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 very similar to glucose with a slight difference. It has a 6-carbon ring like glucose. Fructose has a 5-carbon ring. But they all can be described as C6H12O6. They all have 6 carbons, 12 hydrogens, and 6 oxygen. There we have what are known as disaccharides, which are 2 monosaccharides covalently bonded to each other. Sucrose, maltose, and lactose are the disaccharides. Sucrose is a disaccharide that results from 1 glucose molecule covalently bonded to 1 fructose molecule. Maltose is 2 glucose molecules bonded to each other, and lactose is 1 galactose molecule covalently bonded to 1 glucose molecule. So these simple sugars are the monosaccharides and disaccharides. The complex sugars, glycogen and starch. Glycogen is the storage protein in animals. Starch is the storage form of glucose in plants. Glycogen in animals is stored in the liver. Here's an example of a glycogen molecule. It's not a great example because there's only 3, let's say 1, 2, 3 glucose molecules that make up this glycogen. Glycogen molecules tend to be 2,000 to 60,000 glucose molecules, all covalently bonded to each other. But this is stored within the liver and to a lesser degree within skeletal muscle. Starch looks somewhat similar to this, but since we're talking about human biology, human physiology, we're focusing on glycogen for now. So glycogen is the storage form of glucose. What is the role of insulin again? Insulin, the goal of insulin is to lower blood glucose concentrations or blood glucose levels. That means insulin is going to stimulate the process of pulling glucose out of the blood into hepatocytes, which are liver cells, and it's going to bond these glucose molecules together to create glycogen, and the glycogen is stored in the event that we are low in glucose. What is the role of glucagon? Well, like insulin, glucagon is released from the pancreas, whereas insulin is released from the pancreatic beta cells. Glucagon is released from the pancreatic alpha cells. And glucagon is released during times of low blood glucose levels. And what that is going to cause to happen is the breakdown or hydrolysis of glycogen. It's going to break apart glycogen and put these individual glucose molecules into the blood to increase our blood glucose levels. So once again, insulin is released during times of elevated blood glucose levels to synthesize more glycogen or to facilitate glucose uptake in other cells in the body, insulin-dependent cells. And glucagon is released when there are diminished blood glucose levels and we need to get more glucose into the blood. And that is achieved by breaking down glycogen and putting glucose into the blood. Now let's talk about proteins. Proteins are probably the most ubiquitous biomolecule within the body. If you were asked what type of molecule is X, if you had no idea, say proteins, because the odds are you're going to be correct. So proteins, we've talked about proteins make up channels, enzymes, antibodies, receptors, a whole host of structures within the body. Making up a protein is somewhat complex. It first derives from the DNA housed in the nucleus of our cells. The DNA provides the genetic blueprint for building proteins. And the first thing that's going to happen, I should say, there's 3, maybe 4 structures to a protein. We refer to a primary structure, a secondary structure, a tertiary structure, and then some proteins have a quaternary structure. So the primary structure is the amino acid sequence. Amino acids are building blocks to proteins. There's 20 amino acids in nature that are utilized and put together in different orders to make what's known as a polypeptide chain. This is a polypeptide chain here made up of 5 amino acids: glycine, alanine, tyrosine, lysine, and glutamine. And here's another one: serine, alanine, tyrosine, lysine, and glutamine. So the only difference is the glycine and the serine. And the fact that these amino acids are different and only these 2 amino acids is going to make a very different protein. So once again, the primary structure of proteins is the amino acid sequence that we see right here. These are just 2 examples of amino acid sequences. The secondary structure is how the protein starts developing a shape or morphology. And the 2 basic morphologies of secondary structures of proteins are the alpha helices, or the alpha helix, and the beta pleated sheets. This is an alpha helix, not necessarily a great drawing, but it's hard to draw a 2-dimensional alpha helix. And these are the beta pleated sheets. These red dots in both of these structures are representing hydrogen bonds. So we talked about hydrogen bonds that happen between adjacent water molecules, but they also play a part in proteins as well. So keep in mind, we've talked about pH. If the pH deviates from the acceptable range, it can wreak havoc with the hydrogen bonds in proteins. The tertiary structure of proteins is when that amino acid sequence, after it's developed either the pleated sheet or alpha helix, folds upon itself, and hydrogen bonding plays a part in this as well. You can see some hydrogen bonds here, and this is just, I don't know what protein this is, but I'm just trying to show you how it is folded upon itself. It was initially alpha helix. It still is an alpha helix, but then it folded upon itself. The quaternary structure is when different polypeptide chains come together to form a structure. So one example we have right here is hemoglobin. Hemoglobin is created by 4 polypeptide chains, and there's 4 here. That's not why we call it a quaternary structure. It could be 3, it could be 9. But the quaternary structure is just when a protein is incorporating a number of polypeptide chains within its overall structure. Hemoglobin has what are known as heme groups. There are 4 heme groups within the hemoglobin molecule, and at the center of each heme group is the iron atom. And the iron atom is what oxygen binds to when it is being delivered. Once again, hemoglobin is a molecule within red blood cells that transports oxygen. The great majority of our oxygen is delivered to the tissues bound to hemoglobin. When oxygen binds to the first hemoglobin, it causes a slight conformational change, allowing other oxygen molecules to bind to the other heme groups or the other iron atoms. My point in saying that is that, once again, conformational changes are intended changes in shape of a protein, not to be confused with when a protein denatures. So once again, hemoglobin delivers oxygen. Let's take a look at these enzymes. Once again, enzymes are a type of protein. I should say enzymes are composed of proteins. Enzymes catalyze reactions. They speed up reactions in the body. It's super, super helpful and important to have enzymes that speed up reactions in the body. So here we have an example of 2 glucose molecules being bound to each other via this enzyme right here. Once again, 2 glucose molecules make up the disaccharide maltose. And each of these glucose molecules had a docking station within this enzyme. They sit in that docking station, get right next to each other, and that helps facilitate the covalent bonding of these 2 adjacent glucose molecules. In the absence of an enzyme, it would take time for those glucose molecules to orient themselves in the correct manner so they can form the covalent bond. So it happens much more rapidly and easily in the presence of enzymes, which are catalysts. Now, we talked about denaturing proteins, or when proteins are denatured. When proteins are denatured, it's an unintended change in shape of the protein. And this can happen due to changes in pH. It can happen due to elevated temperatures. It could even happen to differing ion concentrations within the extracellular fluid or intracellular fluid. But let me just show you, here's the same enzyme that has been denatured. And notice the docking station for this glucose molecule right here. This docking station is a half circle, if you will, and this glucose molecule doesn't fit in that half circle. So there's no way it can dock in that spot right there. And there's even no docking station for this glucose molecule. This is due to this protein being denatured, and it's non-functional. So that's why we need to be very mindful of, among other things, pH, temperature, and ion concentrations. Now, back to hemoglobin. Hemoglobin, like I said, delivers oxygen. And there's other things that can disrupt proteins, and that is some sort of mutation within our DNA. Within the hemoglobin molecule, there's a specific primary structure for the amino acid sequence for each of these polypeptides within this molecule. But when there is one change from valine to glutamates, don't worry about where that happens within the amino acid sequence, but just one substitution of one amino acid for another that's incorrect, it causes a change in shape of that red blood cell. So here we see an old type of agricultural tool, grass cutting tool, weed cutting tool, known as a sickle. That's the shape or the blade that you would cut down weeds or grass or hay. The red blood cells typically are ovoid in shape and somewhat biconcave. We'll talk about that more when we get to the cardiovascular system. And these red blood cells squeak through small blood vessels known as capillaries. And these red blood cells tend to be very malleable, so if they get to a tight space, they kind of squeeze and change shape and get through that space. But in someone who has sickle cell anemia, due to that one substitution in the amino acid, their red blood cells now have a sickle shape, hence the term sickle cell anemia. And those cells tend to get stuck within capillary beds and can no longer travel through the capillary beds. And if red blood cells cannot travel through the capillary beds, then they're not delivering oxygen, which is not a good thing. We've talked about carbohydrates and proteins, but we also have lipids and fats. Lipids and fats are basically the same thing. There's different types of lipids. We've talked about phospholipids. There are steroid molecules, which are a type of lipid. All steroid molecules are based off of this molecule right here known as cholesterol. We need cholesterol in our body. There's good cholesterol and bad cholesterol, and we'll talk about that as we move forward. But all steroid molecules are formed off of the molecule cholesterol. Types of steroid molecules, just to name a few, estrogen, testosterone, and cortisol are all steroid-based molecules. Within fats, we also have triglycerides. And keep in mind, when I'm talking lipids and fats, these are nonpolar molecules. And this is a great example showing why it would have the characteristic of nonpolarity or being lipophilic or hydrophobic. This is a triglyceride that is composed of one glycerol molecule and fatty acids. In this case, because it's a triglyceride, it has 3 fatty acids. And like I showed you within the carbohydrates, each of these vertices right here is representing a carbon atom with hydrogens coming off of them. So lipids tend to be carbon and hydrogen. That is to say, there's not a lot of electronegative atoms like oxygen. Certainly we do see some oxygen up here, but it's primarily a hydrocarbon, which confers its characteristic of being nonpolar. The whole conversation of biomolecules would be amazingly incomplete if we didn't talk about this molecule, ATP, adenosine triphosphate. This is composed of one ribose molecule, an adenosine molecule, and 3 phosphates. One thing to keep in mind with ATP, which is a molecule our body uses to do work, each of these phosphate molecules is negatively charged. Because they're negatively charged, they don't really want to be around each other. So it's somewhat like a loaded spring. That is to say, there's a lot of tension within this end of the molecule because of these 3 negative phosphates. As a result, if one of these bonds is broken, this is ATP. If you break one of these bonds, you're going to make the molecule ADP, adenosine diphosphate. But in the breaking of this bond, because this is such an energy-rich molecule, like a loaded spring, that spring's going to take off if you let it take off. You break this bond right here, not only is the phosphate going to take off, it's going to liberate a ton of energy, and that's energy we're going to use to do work. Since we're talking about these biomolecules, we might as well bring it back full circle to the cell. Once again, not all cells are circular. I don't know why I draw them all circular. Here's a cell. This blue structure right here is the nucleus. It's one of the organelles within human body cells. An organelle is a membrane-bound structure with a specific specialized function within the cell. And the function of the nucleus is to house the genetic material, otherwise known as DNA. This right here is the mitochondria. The role of the mitochondria is to synthesize or produce ATP. It is not the only place ATP is produced, but the great majority of our ATP is produced within our mitochondria. Cells that have more mitochondria are going to produce more ATP. Right here we have a ribosome, which officially is not an organelle because it's not a membrane-bound structure, but it is super important in the synthesis of proteins. And while we're at the cell, I just want to point out two structures right here. These surface appendages coming off the cell membrane are microvilli, and the role of microvilli is to increase surface area of a cell. The more surface area a cell has, the more receptors, the more channels it can have, and the more metabolically active it can be. These right here, which tend to look very similar to microvilli, these are cilia, and cilia actually function to propel things away from cells. Certainly they're going to increase the surface area, but that's not really their intended function. Their function is to propel things away from cells. A great example is within our trachea or windpipe, it is lined by an epithelial tissue containing cilia. And also within that epithelial tissue, there's a bunch of mucus, and the mucus is intended to be there to trap any pathogens, dust, debris, or microorganisms that are trying to enter our body via the respiratory system. Those trapped pathogens or microorganisms are going to get propelled superiorly by the cilia, eventually into the oral cavity, which one can eliminate that from their mouth, or they can even swallow it, as gross as that sounds, but we have digestive enzymes within our stomach intended to break that down. And a great example of cilia, or the non-functioning of cilia, is cigarette smokers. Cigarette smokers can get paralyzed cilia, so they're not functioning anymore. And as a result, they have to cough a lot to get that mucus up and out of their airway because their cilia are not functioning anymore. So these are biomolecules and a little bit about our body's cells.